go there. But start at 3 like usual, and it'll be a short thing because Grubau's got somewhere to be at 4. So the time fairy will visit all of us and give us a little bit of extra time. So let's see, Grubau's been talking about nutrient cycles, so I'm gonna keep talking about nutrients but not cycles. And so we'll talk about nutrient use, and then this fun word stoichiometry, which you may remember from chemistry, and it's not exactly like chemistry, but similar to chemistry. So there's that.
Is that a good thing or a bad thing?
It's
A thing?
It's a thing. Depending on how into it you get, it can get a little difficult, but general concept is a good thing. Okay. All right, so these are some boring PowerPoints. I don't have a lot of photos in these, so it's kind of boring, but it's like
An actual book in those photos.
yeah. All right. So nutrients, just to give you a definition, which you've probably already gotten one, element that's required by organisms for growth. And the ones that are generally more interesting are the elements that are potentially limiting to organisms. Did Grubau talk about nutrient limitation at all? A little bit. So out in the world, there's all these nutrients we need to make organisms, and some of them are more readily available than others. The ones that are less available, if they're hard enough to come by, they can limit growth. So, you know how we put fertilizers on plants? Fertilizers are basically made up of nutrients that are limiting. You put on the fertilizer, now the plants have more of that nutrient and they can grow taller, make more seeds, whatever it is. We'll talk more about nutrient limitation here. Common nutrients that we talk about that might be limiting include nitrogen, phosphorus, and silica can be limiting for diatoms in some situations. And iron can be limiting, especially in the ocean. Iron limitation can limit algal growth in the ocean. So, those are just some examples. And so all these nutrient cycles you heard about, there was probably a lot of talk about how autotrophs take up nutrients, and then consumers eat autotrophs, and those elements move through the food web, and then decomposers return those elements back to a form that the autotrophs can take up. Does that sound familiar? Kind of, sort of.
Kind of, sort of.
So autotrophs, land plants or algae or cyanobacteria, they use inorganic versions of nutrients. And so what I mean by inorganic is it's not like a chunk of an organism. Like we use organic nutrients because we're eating like living things, and so our nutrients are coming from organic molecules that were part of a living thing. Inorganic nutrients, what I mean by that is it's out in the environment and it's not obviously connected to some kind of organism. So for example, like nitrogen in inorganic form would be nitrate or nitrite or ammonium, and that would be inorganic, and organic form would be like a protein would have nitrogen in there. So if we're thinking about biological molecules, that's what I mean by organic. It's not really the organic chemistry definition of organic. Okay, but there are some autotrophs that can use a few organic forms of nutrients. Not the land plants, but there are some algae that might be able to take up like proteins or something weird. And then heterotrophs, all of our consumers, they are using organic forms. And it could be actual organisms, or it could be organic, or the biological molecules with that element that are dissolved in water. far so good? Okay. So let's look at, we're gonna talk about nutrient concentrations, so like how much of that nutrient's in the environment. We're gonna talk about uptake. And so that's, our autotrophs participate in uptake. They take things up, and what is happening is uptake. There is not, we don't say uptaking, wait, what am I trying to say? There's a way that people use uptake wrong all the time, and I'm just blanking on what it is right now. I'll get there. We're gonna talk about assimilation, taking, like, uptake is bringing that nutrient into the body, assimilating is turning it into body parts, and then growth rates. And so kind of, the next several slides are gonna be thinking about how the nutrient availability in the environment affects organisms, essentially. And mostly algae and plants like macrophytes, also bacteria and fungi. So all of these organisms can take up nutrients from the environment and not, they don't have to take it up from organisms necessarily. Bacteria and fungi, we think of them mostly as consumers, not cyanobacteria, but heterotrophic bacteria. And they may be eating like other cells or organic molecules, but they might also take up inorganic forms as well. And the study I worked on during my dissertation, we added nutrients to a stream, and the guy that studied the fungi, I told you about this, and the fungi were really stimulated by the nutrients, because they can just take that stuff up. So it's not all about the photosynthetic dudes. All right, and now you're gonna love this. So here's the scary formula that you do not have to learn. You do not have to learn the formula. But there's some take-home messages that I want you to get from the formula. So I just kind of want you to see it and maybe recognize it. And then if you ever go to grad school and do uptake, you'll have seen it before. You may or may not have heard of this in chemistry. The Michaelis-Menten or Monod equation that sometimes comes up in chemistry. Okay, so we have two relationships we want to talk about. This first one is called the Michaelis-Menten relationship. Sometimes you hear about this in chemistry in terms of like enzymes acting on a substrate, but we're thinking about it in terms of uptake rate. So how fast, how quickly autotrophs are taking in an element, and the influence of those external nutrient concentrations. So how fast can algae take up a nutrient? Well, that also depends on how much of it's available outside. And someone did the math. And so I'm just gonna define the parameters of the equation, which, again, this is just for you to understand the take-home messages that are on the next slide. But V here is the just overall uptake rate, so how fast a nutrient's getting taken up by an autotroph. And then the next variable here, Vmax, is the maximum possible uptake rate. And when you see the graph, you'll see where these all fit. S is, and so S is in those squared brackets. So those brackets mean concentration. And concentration of, it's called S because of its substrate, so this is that enzyme substrate historical thing. But for our purposes, that concentration of S is the nutrient that we're looking at. So it could be nitrogen or phosphorus or something else. And then our last variable, case of S, the concentration of the nutrient where the uptake rate is half of the maximum of the uptake, half of the maximum possible uptake rate. Okay, so far so good. I think it'll make sense once we see it in the graph. So here is our graph. So we've got S, your nutrient. So here are increasing concentrations of a nutrient. And we've got our uptake rate on the Y. So we've got V here. And they have a nice dashed line at V max. And they put another dashed line at half of the V max. So the maximum uptake rate, and then half of the maximum uptake rate. And then we have the slopely curve, and K sub S, the nutrient concentration where uptake is the half V max. So that little point right there is where that nutrient concentration is. So, kind of the point of all of this. So this shape of the curve is typically what you see for most nutrients. The concentration might vary, like some things might vary, but that general shape is pretty typical. And what you should get out of it is, in the beginning, as we are at low concentrations and we start increasing our nutrient concentrations, uptake happens, like uptake will increase. Uptake rates increase very quickly when nutrients are low. And then there's going to be a certain point where it kind of levels out and it doesn't really get a whole lot faster in terms of uptake. So if I had a box of donuts and there were 4 donuts in there and I opened the box with 4 donuts, y'all would run in and grab a donut really fast. And maybe if I had more donuts, you'd grab them faster. But if I had a whole giant, like, garbage bag full of donuts, then you wouldn't be eating them any faster just because there was more of them. So, pretend you like donuts. Okay, so uptake gets faster very quickly at low concentrations. Uptake levels kind of max out once we get to higher nutrient concentrations. And we're approaching that Vmax, that maximum uptake rate. May never quite get there.
Oh, it's like a long thing where you never quite reach the maximum?
Yeah, it's one of them asymptote things. Yeah, so it's slowly approaching, but we'll probably not quite get there. And so so these curves, they like to figure these out for different algal species or different plant species or bacteria versus fungi, different organisms. And what we find is that this K sub S value, like here in the example, it's about 0.1. So 0.1 milligrams per liter of whatever nutrient is, for these organisms, that's where it reaches half of the maximum uptake. But that value is going to be different depending on like what species you're looking at, or it could be the same species but the environment's kind of different. We know that things slow down when it gets colder, so temperature at least would have a big effect. We could have all kinds of things affecting that K sub S. But people still try to figure it out anyway. So again, the general shape of the curve would be the same, but some of the values for the X and Y axis may differ for different species. And also when you get into weird things like same species but larger cells, you can also get differences in Vmax or K sub S. Alright, so that's the Michaelis-Menten. Michaelis-Menten is uptake rate compared to its concentration in the environment. And then our other equation looks very similar. And it also starts with an M, but instead of being about uptake rate, it's about growth of the organisms. So now we have the Minode equation. And so this is the relationship between external nutrient concentration, so same as Michaelis-Menten, but it's the relationship with growth instead of uptake. And the formula looks exactly the same, and except instead of talking about uptake, we talk about growth in that BU thing there. And then the same kind of take-home messages is growth rate will increase rapidly when you're at low nutrient concentrations, and then it's going to kind of max out once you approach a maximum growth rate. Like there's only so fast a cell can grow. Unless they're exposed to radiation and they get superpowers. And then, just like with the Michaelis-Menten, all these values are gonna be different depending on like which algal taxa you're talking about, if there's a difference in cell size, all that stuff. So when we're just kind of generally thinking about like how are nutrient concentrations affecting algae, we might want to know about how fast they can take it up or how fast they're growing, depending on how much nutrient is available to them. All right, was that fun or what? Good stuff? Okay, that was the most painful part of the day. All right, now that's over with. Questions, comments, concerns? Okay. So, still thinking about uptake rate and growth rate. So just in general, this will be a function of how much nutrients are available, for sure. Also some other things like temperature. Life slows down when it's colder, so everything slows down when it's colder. Light, I say remember the PI curves. I haven't shown you those yet, so you don't remember those. I'll show you those in a second. pH or some kind of other conditions can also affect uptake and growth. And so I'll show you the PI in a second. So these curves here, it's showing uptake of nitrate. This graph is uptake of nitrate compared to how much light is available. And you see a similar shape where that uptake increases rapidly at low light concentrations, but at some point it's gonna max out, and adding more light doesn't make them take up nitrate any faster. A very similar relationship, I think this is supposed to be ammonium. Goes up really fast at low light as light increases, but then it maxes out. And then, so that's light affecting uptake. This is an example of an other interaction where you've got nitrate uptake compared to how much ammonium is available. And so when ammonium is low, uptake of nitrate is higher. As ammonium increases, that's preferred by algae, so they'll take up less nitrate if they can get ammonium instead. So there's all kinds of things to complicate life. And I know you're dying to see this PI curve. Look at the PI curve. So exciting. So, back when I taught this class by myself, and I was doing the chemistry, this was in the intro to chemistry. Come on. and I don't want to spend a ton of time on this, but PI refers to photosynthesis and irradiance. Irradiance is just how much light is available. And so it's similar to that Michaelis Menten and Minode when we're looking at nutrient concentrations and how that affects uptake and growth. This is looking at photosynthetic rates compared to how much light is available. And we see similar shapes in these curves where we've got light on the X and photosynthesis on the Y, and at low light levels, when we increase light, we increase photosynthesis really quickly. At some point we max out though, and actually if light gets too high, photosynthesis will get reduced. We talk about something called the compensation point, and this is where, so this net photosynthesis, we've got 0 up here and -1 down here. Does that ring a bell from our all-night party on the dock with the probes and the BOD bottles, all that? So below 0, where we have more more respiration than photosynthesis. Above 0, more photosynthesis than respiration. So that compensation point is 0, where they're equal. And so that would be like the light level where that happens might be of interest. And the only other thing I want to mention, I don't really want to talk about Pmax, it's just the maximum photosynthetic rate you get, like what light level that is. But photoinhibition is a thing that I wanted to mention, where if light levels get too high, so if you imagine a photosynthetic organism with their little chlorophyll molecules out there and they're grabbing onto electrons, or they're grabbing onto photons, sorry, and capturing that energy. So when light is low, those molecules aren't getting like maximally used. There's plenty of chlorophyll available to catch photons. As we increase, all those molecules get kind of full of photons, and so they can't capture any more. And if you add even more light, it'll slow them down. And so there's going to be some point at which, and this happens for plants, it happens for algae, where there's just like too much light and they can't photosynthesize anymore, and they're actually kind of impaired by it a little bit. So photoinhibition is a thing. But again, similar shape, things speed up at low levels of some resource. They speed up really quickly as you add more of that resource when it's in short supply. And then it maxes out. Alright, does that make sense? Alright, moving on. Okay. So, no graphs here. Just some other ideas I wanted to talk about. The concept of luxury consumption. Doesn't that sound exciting? And sometimes certain organisms are able to take up more nutrients than they actually need to use at any one time, and they can store them and use them later. So when we store energy in our fat cells, that's luxury consumption. If we have more food available, we can take in that extra food, and some of that extra energy will get stored, and we can use it later if we're low on food. And so some organisms can, for example, take up extra phosphorus and store it in special structures, and then if they're short on phosphorus in the environment later, they can just use what they've stored. And so that's a good thing to know about in terms of nutrient uptake, that some organisms do that. Different topic entirely, macrophytes are aquatic plants, so just some weird things to keep in mind when we're thinking about nutrients and macrophytes is that they can get, so where do plants normally get their nutrients from?
On the ground.
The ground, yeah, out of their roots, right? Macrophytes being in the water, they can also get some from the water column. So macrophytes can get nutrients through their roots from the sediment. They can also get it from the water column. And if you're trying to figure out where they're getting those elements, it's not easy to do that. Next slide we'll talk about a way that you can do that. But my roommate in grad school, she was looking at an aquatic plant called Potamogeton, and she wanted to know how algal epiphytes growing on the plant would affect it. And so she was curious about this question of nutrients coming from the soil or from the water column. If you've got a lot of algae growing on your leaves, are the algae stealing all the nutrients out of the water column and you're using more out of the sediment? She was curious about that kind of thing. And so really the only way to figure something like that out is to do what we call a tracer experiment. And tracers are when we find some radioactive isotopes of our element of interest. And for nutrients, like in the macrophytes, we would get some radioactive nitrogen and maybe squirt it in the soil and get some radioactive phosphorus and squirt it in the soil. And then let the plants do their thing for a little bit and then sample the plant and try to see if there's any radioactive atoms in there. And so it's not like super duper radioactive, it's a relatively safe amount of radioactivity, but it's still radioactivity. But that would be a way to figure that out, if macrophytes are getting, like how much of their nutrients are from the soil versus the water column. So tracers can help you with that. Other things you can do with tracers is like figure out how quickly nutrients are moving through an organism. So kind of the balance between uptake, taking something up, that's what I was going to complain about. The verb is to take up, not uptaking. So you wouldn't say the plant is uptaking nitrogen, you would say the plant is taking up nitrogen. That's my pet peeve. Okay, so, but this nutrient availability, looking at the balance between taking something up and then the remineralization part where it's going from, so uptake is going from the environment into an organism. Remineralization is when you're going from an organism back to the environment. So it's that like decomposition or just release back to the environment. And so tracers let you figure out like where nutrients are coming from that get into organisms, because you create this radioactive source of a nutrient and see where it goes. And then you can also find out if there's other sources that are not radioactive that are still getting in there. Main point, radioactive isotopes called tracers can help you figure out where nutrients originate for organisms. All right. Y'all doing all right? Okay. Gonna get some pictures soon. Yay. So let's talk about nutrient limitation a little bit more. Because one of our big human impacts on aquatic ecosystems is sort of releasing a lot of organisms from nutrient limitation because we've added so much fertilizer to the environment. There's a lot more nitrogen and phosphorus out there, and that has changed this nutrient limitation problem. So nutrient limitation is the control of growth or productivity or production by a nutrient, or it could be more than one nutrient. I could grow more if I only had more fill in the blank. More nitrogen, more phosphorus. This is also known as Liebig's law of the minimum. And Liebig's law says that the rate of a process, like growth or decomposition, will be limited by the rate of its slowest subprocess. So, it's a really boring example, but when I photocopy my Biology 120 exams, I've got an answer sheet and I've got the test, and I print all the answer sheets, and then I set the copier to print all the tests, and as the tests come out, I stick the answer sheet in there. And I go faster than the copier goes. I can stuff them in there faster than the copier can make more exams, and so I'm limited by how fast that copier goes for the whole thing to get done. So I'm sitting around waiting for that copier. So there's always some process that all the other processes will be sitting around waiting for this other process to get its act together and do what it needs to do. And so that's one part of the Liebig's law of the minimum idea, and the other that goes with that is organisms are limited by resources. So because of that, the rate of a process limited by its slowest subprocess, the organisms are limited by resources that are in shortest supply relative to its needs. So compared to all the other resources it needs, that one thing that there's not quite enough of is going to be what's limiting growth or reproduction or whatever. So if you were building cars, for example, you would need, each car needs a steering wheel and 4 tires and an engine, et cetera, et cetera. And it doesn't matter if you had 1 million steering wheels, if you run out of engines, you can't make more cars until you have more engines. So that relative to its needs, that's the big thing. It doesn't matter how much other stuff you have when there's one thing that you just don't have very much of. I also think of it when you need to do laundry, there's that one article of clothing that you are in short supply. Like you always run out of socks before anything else, but you have to do laundry when you run out of socks, even though you have more pants. Because you just need clean socks. So there's that. So we good with that? Does that make sense? Okay. Picture time. So how do we figure out what nutrients are limiting in aquatic ecosystems? And there's fun experiments that were a lot of people were doing these like back in the 70s and 80s and probably early 90s. Pretty straightforward, but basically we add nutrients and look at what happens. So there's, we can make what are referred to as mesocosms. And mesocosms are like miniature versions of that whole ecosystem. So we look at this photo. So here we are in a big lake, and on this raft, you've got basically giant plastic bags with lake water on the inside. And so it's like a little miniature lake in there. And then you can add nutrients to it and see what the plankton do. And so it's water from the lake, it's gonna be at the same temperature, it will get the same light kinda, so it's similar-ish to the lake itself. You don't really get the water flow you'd get normally, it's not perfect. But you could add nutrients and see which ones spike algal growth, and then that'll tell you what's limiting. These things, so mesocosm is one term, those things have also been called limno corrals, so that's just a fun term. We've also done assays, and this flower pot here is not a flower pot, it's an NDS, a nutrient diffusing substratum. And so what's going on with that flower pot is the inside of the flower pot is filled with agar that has nutrients dissolved in it, and those nutrients ooze out of the porous makeup of that flower pot, and you've got that underwater, and so algae will grow on the outside of the flower pot. And if they're limited by phosphorus, say, in that ecosystem, you'd see a lot of algal growth on that phosphorus flower pot. So, and there's different, I've got another picture of some other nutrient diffusing substrata. So basically creating sort of artificial environments, adding nutrients and seeing which ones evoke the most response. And there's a lot of criticisms with these methods, like nutrients don't usually ooze out of rocks, they generally come from the water column. Those Limno corrals are not exactly like the real lake, and so it's not perfect, but it can still tell you some things about what nutrients might be limiting in those systems. And typically what we see in freshwater is nitrogen is limiting, or phosphorus is limiting, or it's actually both of them together that are limiting. So if you add, when both of them are limiting, you could add nitrogen by itself and not see a response. You could add phosphorus by itself and not see a response, but when you add them together, you see a response. So that can happen. Depending on the ecosystem, you might even see like carbon limitation or silica or iron as well. Okay. So I wish this were a color picture, but it is not. So these are these weird kind of screw top plastic bags, and they had lake water in them with nutrients added, and I think they were probably floating in the water in some way. But you can see like this one is a lot darker than all the others. That's probably green. There's probably a bunch of algae growing in there. So whatever nutrients they added to that one really stimulated algal growth. In streams, those flower pots could be used in streams or in lakes. These could be used in streams or lakes, so what they've done here is, do you remember film canisters? So back when we had to go buy film to put in our cameras, the film roll would come in these plastic cylinders.
With the cap.
With the gray cap, and they were black and they had the gray cap. So if you imagine like a container like that where you take the lid and cut a hole in it, and inside of the container you would have the nutrient agar stuff, and then you'd put a piece of filter paper on top and then put the cap on top. So the hole in the cap would let that filter paper be exposed to water, and then the nutrients would ooze out from the agar, and then you get algae growing on the filter paper. So these are a bunch of film canisters that are attached to something and then put out in a stream, and you can see that there's algae growing on these. And as any good scientist would have, they've got a bunch of replication here. So they pulled the filters out and put them in these centrifuge tubes. And so what it looks like, here's our control filter paper with no nutrients added. Here's the ones where nitrogen was added. Here's the one where phosphorus was added, and here we had nitrogen and phosphorus together. So there's a little bit of algae at least growing on everything, but which nutrient or nutrients look like they're the most limiting?
Nitrogen and phosphorus together.
You have a vote for both of them together? Y'all agree? I would say yes. So not a whole lot of stimulation with nitrogen by itself, not a whole lot of stimulation with phosphorus by itself. Like, these two columns look a lot like the control column, but when you've got them both together, you're seeing a little bit more algal growth there. And so you can actually measure the pigments and quantify your response there.
So they're both almost, in some ecosystems, there's both some, like not each other's, but like equally the limiting.
Let me show you the next graph. It will tell you. So, this is showing, just that question. So, this is lakes, wetlands, and streams. Someone went and looked at a bunch of old papers and tried to, and basically quantified how many were nitrogen limited only, not phosphorus, how many were phosphorus limited only, not nitrogen, how many were both. They also did potassium, which can be, it's more of a terrestrial limiting nutrient. But some places there was no nutrient limitation, a few it was just nitrogen, so like in lakes, a few were just nitrogen, more were phosphorus, but most of them were both. Streams were fairly similar, and then some wetlands, not a lot of N and P, and more when you add in the potassium question. So it just depends on the location and the geology and the history and all that stuff. Human impact. Alright, how cool is that? So cool. Let's see, stoichiometry. Let's see how far we can get. Okay, so stoichiometry, what we're talking about in ecology is the relative ratio of nutrients that we find in organisms. So how many carbon atoms to how many nitrogen atoms to how many phosphorus atoms do we typically see? And we talk about the Redfield ratio. Anybody ever heard of the Redfield ratio? A lot of names today. Michaelis Menten, Minot, Liebig, Redfield. So, on average, across a whole bunch of different organisms, we tend to see a ratio of carbon to nitrogen to phosphorus that's 106 to 16 to 1. So for every 106 carbon atoms in an organism, we tend to see 16 nitrogen atoms and 1 phosphorus atom. On average. So if you went and counted all of your atoms, you might not be exactly that, but you might be in the general ballpark. If you compare different species, they might be a little bit different.
If it wasn't dependent on how much was in the environment, you would think carbon would be the most limiting because it's the most needed.
Mm-hmm. But there's so much
abundance.
Yeah. Carbon. That ratio, it can vary, again, like I said, by species, by taxon, or by physiological state. So, like if you're trying to accumulate resources for winter, you might, or if you're like getting into a hibernation state, you might have a bunch of extra nutrients available to deal with that, for example. Or if you're starving, that might be different as well. But we sometimes use this ratio to try to determine or understand nutrient limitation. So without doing all the experiments where you're growing algae and trying to see what might be limiting, you could just go out, take a water sample, measure nitrogen and phosphorus and see what the ratio is. And so if you've got like 1,000 nitrogen atoms for 1 phosphorus atom, you can probably guess that you are not limited by nitrogen because there's so much of it. It's probably phosphorus that's limiting. So there's that. And this table, I want you to focus on this column on the right. And so we've got a bunch of different elements and how much of these elements we think are in plants and algae and how much are in the rivers. And then there's this demand compared to supply. And so what this is saying is that the number that's in this column, the smaller the number is, that means the easier it is for the organism to get that particular element. So hydrogen and oxygen, why is that so easy for aquatic organisms to get?
No clue.
H2O, so much less than 1, a lot of it available. When we look at the largest numbers here, we've got nitrogen and phosphorus. So that's no surprise, those two are the typically limiting nutrients. All right, there's a little bit more about, we have a few more slides, but I'm done for today. So we'll get to those another time.
3 slides.
Huh?